Okay, guys, it is that time. We have one more judge to wait on, but we'll see if they can make it soon. So my name is Parker Westover. Welcome to the Environmental Science Division. We have a total of 4 presentations in this division. At the end of each presentation, we'll take questions from the judges and then the audience. You may ask your questions through the chat or use the Zoom raise hand feature and then wait to be called on. Before we begin this presentation, can the judges please briefly introduce yourself and mention your research field, please? That'd be great.
Sure, I guess I'll start. My name's Ben. I'm a PhD student. Oh, are you wanting us to just say it or in the chat?
Yeah, you can go ahead and say it. That'd be great.
Yeah, so I'm a PhD student at Baylor. I'm technically in the biomedical studies, the Institute of Biomedical Studies, but my advisor's in the environmental science department, and we work with like artificial ecosystems like streams and wetlands and big experimental ecosystems. And what I study are fate and transport and bioavailability of nanoparticles, kind of in like under realistic exposure scenarios.
Awesome, thank you so much.
And then I guess it's me. So my name's Ashley Ball. I'm also in the environmental science department at Baylor. I'm in Dr. Lovato's lab, so we do animal alternative modeling. So my work is predominantly with comparative studies at the moment, looking at human responses, fish responses, and bird responses, all to chemicals that have been largely previously studied in vivo, but we're trying to get a gauge on how the models respond when it's not a mammalian model.
Awesome. Thank you guys for being here. So right now we will begin with our first presentation. We have Alec Kramer from Baylor University. You may share your screen and begin whenever you are ready.
Cool, fantastic. All right, can everyone see my screen?
Yes.
Yes, okay. Need the confirmation there. All right. So I've had $1 for every time I struggled to get my screen share in a Zoom chat. All right, here we are. Everyone should be able to see that. And can you all also see my mouse on the screen? All right, I'm going to take that as a yes then. So, hello everyone, thank you all for coming out today. My name is Alec Kramer, and I'm going to be presenting on my research over the effects of magnesium sulfate pretreatment on induced neurotoxicity. And this is a population and functional recovery study of neuroblastoma in vitro models. My name is Alec Kramer, and I worked on this project in partnership with Saher H. Pradam and Dr. Christy M. Sayes at Baylor University. To begin my presentation today, I'd like to speak about the relationship between Parkinson's disease and magnesium deficiency. Now, Parkinson's disease is a neurodegenerative disorder that impacts dopaminergic neurons within the substantia nigra region of the brain. A clinical association has been found within several studies between low levels of magnesium within the central nervous system and the progression of Parkinson's disease. Now, magnesium is an essential cation that plays a role in many physiological functions, and additionally, magnesium deficiency in adults shows a non-specific pattern of progression that impacts both the central nervous system and the peripheral nervous system. In this study, we utilized 2 different neurotoxicants for a comparative analysis. We utilized 6-hydroxydopamine, and for the purposes of this presentation, that's been abbreviated to 6-ODHA on the slides, and that is its abbreviation found commonly in literature. 6-hydroxydopamine is a common neurotoxin that is used to induce a Parkinson's-like effect on cellular models. It's used to study the biochemical mechanisms of Parkinson's disease, especially with its autooxidative properties, its selective targeting of dopaminergic neurons, and its ability to induce cellular stress. Lead is our environmentally relevant neurotoxin that we utilize in this study. It is commonly contacted through occupational hazards as well as within fuel byproducts. Now, lead does have the capacity to cross the blood-brain barrier, and it's through this mimicry of calcium that's able to be inducted into the central nervous system. And once inside the central nervous system, lead is able to accumulate within motor regions of the brain, such as the substantia nigra. And it's because Parkinson's disease and this lead exposure affects similar physiological functions and structures within the brain that these were used for the comparison and the comparative analysis within this study. So the purpose of our experiment today is to assess the long-term recovery of neuronal cells after exposure to a neurotoxicant environment following a magnesium sulfate pretreatment. Now, this study was divided up into roughly 3 phases. We had a pretreatment phase where cells were incubated with magnesium sulfate at 1 micromolar concentration for 1 hour. And this constituted our pretreatment phase. Following this pretreatment, they were washed and then they began their acute exposure phase. And this is where cells were incubated with either neurotoxicant for 24 hours at the LC50 concentrations. And so for 6-hydroxydopamine, that concentration was 50 micromolars. And for lead chloride, that concentration was 0.1 millimolars. However, in order to determine the recovery after exposure, the cells were washed with PBS and then returned into just a plain media environment where they were allowed to grow for 24, 48, and 72 hours. And at these time points, the cells were measured for viability, ROS generation, and an inflammatory response. Moving on to the results of our study, we utilized a Presto Blue assay to assess cellular viability and cell recovery. Looking at our graphs here, we can see that the 6-hydroxydopamine and lead chloride similarly reacted within these environments. Additionally, what we see on both of these graphs here is that the pretreatment does have a protective effect against either toxicant, shown by the improved viability within both exposure scenarios. Also, what we see with the magnesium sulfate 6-hydroxydopamine values on the left graph here is that there is an increased proliferation between the 0 and 72 hours of recovery, shown by the increases in values on the graph. Additionally, something that we see with these graphs here is that the LC50 exposure of either toxicant led to no or minimal recovery, and that's indicated by this very flat line at the bottom of the graphs here. And this potentially indicates that there is cellular damage happening within these cells that is limiting their capacity to proliferate. Moving on then to our cellular stress, we measured concentrations of the generation of ROS. ROS stands for reactive oxygen species, and an increase of ROS would lead to oxidative stress within the cellular environment. This data here is normalized to the control, so that value 1 with the red line running across the stream is representative of our untreated control group. What we see within this data is that over time, ROS generation is decreased within all exposure groups and with all exposure scenarios. However, what we see significantly is that there is an increase in ROS generation within the LC50 concentrations of 6-hydroxydopamine and lead chloride exposure. And that appears to be almost twice that of the regular control. Additionally, looking at this graph, we see that magnesium sulfate pretreatment in both the lead chloride and the 6-hydroxydopamine exposure groups is able to reduce the ROS generation significantly just by the pretreatment alone. Continuing on, looking at our inflammatory response, we measured expressions and concentrations of interleukin-6. IL-6 is a protein that's generated and it's a high indication of an inflammatory reaction within these cells. So what we see is that there's an ongoing inflammatory stress for these cells between the 0 and the 72 hours of exposure, or post-exposure, my bad. Over time, IL-6 expression is reduced for all exposure scenarios, except for in the magnesium sulfate lead chloride exposure, where we actually see an increase in inflammatory response from the 0 to the 72 hours post-exposure. Also, what we see in our data here is that the 6-hydroxydopamine and lead chloride seem to double the interleukin-6 expression when compared to the control, the untreated control groups. Additionally, looking at the graph here, we see that magnesium sulfate pretreatment is able to aid in the reduction of IL-6 expression for both the 6-hydroxydopamine and the lead chloride exposure. And although the magnesium sulfate followed by the lead chloride exposure seems to increase its inflammatory response from the 0 to the 72 hours, this is still collectively less than that of the lead chloride exposure without the pretreatment. All right, continuing on to some of our microscopy images of these cells, to kind of orient yourselves on these slides here, we have white arrows pointing towards apoptotic cells. These are cells that have died off due to the exposure. We also have areas of green and green arrows pointing towards pointing towards liposome recruitments and lysosome recruitments, my bad. As well as ROS generation as indicated by the red on these slides. And there's quite a bit of red throughout these images here, and that's all indicating towards ROS generation. However, something to note and something of interest within these slides and these images here is within the orange circles, we see a co-localization of ROS generation and lysosomal recruitments. And this is highly indicating that mitophagy is undergoing in these areas. And mitophagy is a cellular process where mitochondria are selectively degraded due to either dysfunction or dysregulation, where the cells are able to go through and remove these mitochondria that are not functioning properly. Now, mitophagy is often utilized as a response to cellular response or to cellular stress. So what we see in a comparison from the 0-hour post-exposure to the 72-hour post-exposure is that in the 0 hour, we see high amounts of lysosomal recruitments, as well as high amounts of ROS generation. And we see quite a few areas where these are happening together. So it's that co-localization of ROS generation and lysosomal recruitments, where we have a high indicating factors that lysosome or that mitophagy is happening in those areas. Following that, looking at our 72 hours of recovery for just a comparison, we still see areas where mitophagy is undergone, and we still see some lysosomal recruitments, but that's relatively low compared to the 72 hours of post-exposure. What we do see is still a high amount of ROS that's generated, but collectively it appears that these cells are under less stress, and it's potentially that mitophagy is leading towards that lowered stress. Also, if I forgot to mention, this slide right here is showing our 6-hydroxydopamine exposure, and the following slide here is showing the lead chloride exposure. And so here, comparing our 0-hour post-exposure of lead chloride to the 72 hours of exposure, what we again see in the 0 hour is that immediately after exposure, we see high amounts of lysosomal recruitments and ROS generation, indicating that these cells also are undergoing mitophagy to try and alleviate some of their cellular stress brought on by the lead chloride exposure. Also, what we see is quite a high amount of apoptotic cells, and that is going to continue on in a trend. So over that 72-hour period, in comparison, we still see quite a bit of ROS generation, and we still see some lysosomal recruitments and some indication that mitophagy is still undergoing in these cells. But a bigger thing to notice within the 72 hours post-exposure images here is that there is a very low cell count, potentially indicating that there is severe damage happening within these cells, causing them to not proliferate. And this would again link back to our Viability study, where we did not see an increase in proliferation caused or within the lead chloride exposure. Moving on then to some of our conclusions of this data here today, is that we conclude that magnesium sulfate did improve cell viability, it decreased the ROS generation, and it decreased interleukin-6 concentrations and expression. Collectively, this data does indicate that the pretreatment of magnesium sulfate was effective in alleviating cellular stress, brought on both in a Parkinson's disease biomimicry scenario and in an environmentally relevant neurotoxicant scenario. Additionally, something else that we see is that within the 72 hours of recovery, it's observed that within each exposure group, the ROS generation and IL-6 concentrations decrease. Of course, with the exception of the IL-6 concentration of the magnesium sulfate followed by the lead chloride exposure. Now, this we can work to conclude that following an acute exposure, there are intrinsic cellular mechanisms that will function to continually alleviate damages brought about by exposure scenarios. One of these mentioned is mitophagy. And again, with our microscopy images, it's highly indicating that ROS generation and lysosomal recruitments, with those two factors together, that these cells are utilizing mitophagy in an attempt to alleviate cellular stress. All right. Totally sorry. All right, moving on then into our future work. Some things that we're going to be looking towards moving forward is going to be rerunning this data to continue assessing trends that we see and to potentially increase our statistical significance of our findings. Also looking forward, we are planning on adding a mitostress assay utilizing an Agilent Seahorse XF analyzer. And this machine and this type of assay is used to assess mitochondrial stress. And with this, we're going to be assessing the influence of mitochondrial dysregulation on cellular recovery. To the right here, we do have some preliminary data from our results from this test. And basically what we can see here is that within our untreated control group, we see high amounts of oxygen consumption, indicating that these mitochondria are high functioning and working at a very high level as they should be. Again, what we see at the bottom of this graph is that we see with the 6-hydroxydopamine exposure is that there's a lower amounts of oxygen consumption, indicating that these mitochondria are under stress. They are not functioning properly as they should. And then in the middle, we see that the magnesium sulfate pretreatment is able to increase the oxygen consumption, indicating that magnesium sulfate is able to alleviate some of the stress that these mitochondria are under, allowing them to function at a higher level. Also, as indicated within our data and within our microscopy, is that further research is going to be needed into the lead chloride exposure to further elucidate some of the mechanisms leading to the cell death that we saw. And lastly, for our future research, this project leaves potential for a co-cultural model to be utilized to try and increase the physiological relevancy of this study. And lastly, for my acknowledgements, I'd like to thank Baylor University's Department of Environmental Science, as well as the Stays Group at Baylor University. I'd also like to thank BURST and HOT for hosting this conference and everyone involved with the events today. And with that, I am available for questions.
Awesome. Yeah, if any judges have any questions, please feel free to ask them. We'll let them ask these questions first, and then we'll go to the general population here. Thank you, Alec, for your time.
Oh, yes, sorry. I have one question in the chat from Benjamin. Yes. All right. So have we considered lowering the concentrations than LC50? And this one, we did not consider lowering the concentrations. We really wanted to see just basically at more of an extreme environment, what would potentially happen with this exposure and with the long-term recovery brought about with the magnesium sulfate. But that's an excellent question. Also, along with the recovery, longer than 72 hours, we chose just the 72 hours just as a basic, almost like a chronic recovery stage. But moving forward, we definitely could expand that out to something like 96 hours or even 120 hours going through 5 days, trying to see if we continue to see trends. Also, what do you hypothesize the effects of magnesium sulfate? All right, so what I would hypothesize the effects of magnesium sulfate would be under a lower dose, but with a longer exposure period. So I would, under a lower dose, I'm assuming referring towards the lower dose of the toxicants that we utilize, and with a longer exposure scenario, I think something that was telling that I saw in the data here, yes, towards this one, especially with the ROS generation, is we saw a significant decrease in the ROS generation with the magnesium sulfate just within the 3-day time frame that we ran the study. And I believe that if we continued, I would hypothesize that we could even at 5 days potentially reduce that ROS generation down to even the baseline control of what we see. And I believe that there's another question in the chat. Carrier solvent for our cellular exposure. So our carrier solvent was just DMEM. So what we did is we just measured our chemicals and we measured our concentrations just to directly put into the cell media, where then we just dosed them within a 96 well plate. Did we do any statistical analysis on this data? We did some statistical analysis on this data. However, that was not entirely complete by the time that the poster presentation has been, by the presentation's time. However, that will be included in our manuscript that we are planning to include this into.
Yes. Hi. Thank you so much for your talk. It was really, really excellent.
Thank you.
I was just wondering, after she asked the question about the carrier solvent, it made me start thinking. So you said you used DMEM, you just dosed the DMEM. Did you use FBS in that at all?
We did not use FBS in that at all.
Okay.
So we didn't want to potentially spike the cells or have an odd increase in the growth rates that wouldn't be controlled by us. So we dose all these cells at just a very consistent confluency at, I think, 80,000 cells per milliliter. So
was that the only thing that you took out of the regular media formulation?
I believe so, but I'm not entirely certain on that. But I know that we did not increase any FBS or anything to try and stimulate growth within these cells.
Okay, and then could you go back to your LC50 slide for me, please?
To which one?
Your your data with the LC50.
Oh, yes. The viability slide?
Yeah, sorry.
No problem, no problem.
Okay. So I think that's really clean data, actually. Very well done. So how did you derive your LC50 then? Like, did you use Probit?
So our LC50 we found through literature sources. So we saw several papers that indicate within the 24-hour time frame that the LC50 value of the 50 micromolar and for the 6-ODHA, and then the LC50 value for the lead chloride was at 0.1 millimolar. And so that's just what we based our study off of, was just previous research that we had found.
Okay, all right.
Mhm.
Cool. Thank
Yeah,
you.
and on those same papers, that is where we found the 1 micromolar concentration of magnesium sulfate as well.
Okay, thank you.
Yep.
Awesome. Thank you so much, Alec. Appreciate your time.
Yes.
We will now present the second presentation. This presentation is by Jake Dickinson and Denise Stevens from Baylor University. And y'all can begin to share your screen whenever you are ready.
Can you guys all see my screen right now?
Yes.
Okay. Hey everyone, my name is Jake Dickinson. I'm a junior honor student majoring in biochemistry who intends to matriculate into medical school after my graduation spring of 2022. Today I'm presenting this with my colleague, Denise Stevens.
My name is Denise Stevens. I am a junior chemistry major planning on attending medical school after graduation in December of 2021. Our talk today is titled Production of Nanocarriers for Agricultural and Environmental Applications.
Previously, our team has presented this work at regional and national conferences such as the Society of Toxicology and the American Chemical Society. Here's a poster I was able to present in the 2020 Fall Lone Star Regional Chapter of the Society of Toxicology last semester. For today's talk, Denny and I will present these topics one at a time in more detail.
I was brought onto this project in January. When I arrived, the lab had previously produced silver, copper, and zinc nanoparticles. These materials are produced using a seeded growth method, which I will explain later. Since my time in lab, I have helped develop the SOP and the synthesis scheme for producing iron particles. Each of these particles are of interest to the United States Department of Agriculture for a variety of reasons. Our interest is to develop stable colloidal particle systems for targeted drug delivery to crops and animals succumb to disease. The USDA is also interested in metallic particles that leach cations into the surrounding soil. Sometimes these are called micronutrients. Lastly, all of the particles have shown antibacterial or bacterial biofilm inhibition properties, which is of critical need to the USDA in both crop and animal production. This slide shows the synthesis scheme for which we use in lab. Silver, copper, iron, and zinc nanoparticles are synthesized using a chemical reduction and capping mechanism based off the Brust-Shiffrin method. The Brust method is derived from a chemistry paper authored by Brust over 2 decades ago, and it describes the gold standard for producing metal colloids in aqueous suspensions. In addition to being the gold standard, it also makes gold nanoparticles. This method produced nanoparticles using a bottoms-up approach. Bottoms-up refers to growing a symmetrical and spherical colloid from an ionic seed to a stable and self-assembled nanoparticle. Alternatively, a top-down approach uses grinding or milling techniques to erode large particles into small entities. To the right, we show the general reaction scheme to produce the stabilized colloids. We start with a metal precursor represented by AB. After some vigorous stirring, the precursor dissociates into its cation and anion forms. After adding some heat and a reducing agent, the cation converts into a zero-valent metal. A capping or stabilizing agent is then added to help promote atomic clustering until eventually producing nanoparticles.
Okay. As previously stated, we produce many different types of nanoparticles such as copper, silver, zinc, and iron. Each of these has to be coated with a stabilizing agent, which will impart properties such as color or charge. Following synthesis, we have to characterize these nanoparticles to ensure that the nanoparticles we produce possess the properties we want. So during the first pass of characterization, we check the color, size, charge, and dispersity. So the pictures on the left show how the nanoparticle's surface coating can impart a variety of colors. Now, if we look to the right in the chart, we can see in the diameter column that there are different sizes for these different nanoparticles and coatings. So while the metal particle itself is under 20 nanometers in diameter, the coating may make it grow to be a little bit larger. For the charge, the coating can impart different charges. Often we use surfactants to confer a positive charge, an acid to confer a negative surface charge. And although these nanoparticles prefer to have a surface charge rather than exist with a neutrally charged surface, we can often use polymers to confer a neutral charge. Note, I say often, because as you can see in this chart with the highlighted PVP nanoparticles, sometimes some of them are really close to neutral, while others are just very negative. So that's something interesting. And dispersity tells us how uniform our particles are. The smaller the dispersity index, the more monodispersed the particles.
As part of this project, we get trained and get to use a variety of analytical techniques too. Here we show just a taste of the type of data collected for the particles. TEM is transmission electron microscopy. These images give us information on the size and shape of the particles synthesized. AFM is atomic force microscopy. These images reveal the topography of the particles. Spectroscopic data is collected too. In fact, I'm currently working with Professor Janae Siren in the chemistry department at Baylor University to collect additional UV-Vis data sets. Here we show FTIR spectra of two of the particles routinely used in lab. FTIR is used to confirm that the nanoparticle system contains both the metal and the surface coating.
Due to just the beauty of these atomic force micrographs, I felt as if we must highlight them. These images were recently captured by Dr. Say's postdoctoral student, Dr. Vicky Liu. The first row shows the first column shows a two-dimensional image of the particles. And the gradient bar, which is shown on the right of everything, shows just how tall and how far they come out in the z-axis. The brighter the color in the image, the taller the nanoparticle. Here in the top one, which is the C-tab coated, the highest, so the brightest area is 175 nanometers, but only in the bottom, it is only 34 nanometers. The other four particles, pictures you see show a three-dimensional images of the particles. Here we show a full characterization slide for one single nanoparticle colloid. In this case, it is a C-tab coated silver nanoparticle. As you can see, we include the size, the charge, the dispersity, the color, shape, and we have an estimated concentration. Additionally, we show UV-Vis, FTIR, atomic force microscopy, and TEM.
In a recent study, we wanted to know if the DLS patterns of each of the three different surface-coated iron nanoparticles change if the particle suspension was concentrated. We synthesized the particles the same way as previously described. We then used a series of centrifugation techniques to concentrate the sample 4 times its original as synthesized product. Overall, we found that the 1x values for dispersity, diameter, and surface charge remain the same for each of the particle systems with different coatings. The images to the left show photographs of the as synthesized or 1x concentrated samples, as well as their corresponding TEM images. The high dispersity index coupled with the TEM images of the polymer-coated iron nanoparticles show that the size population is not uniform, meaning there are a lot of different sized particles in the black solution. On the other hand, the yellow solution shows the lowest average size and relative monodispersity as compared to the black suspension. For surface charge, the acid conferred a negative charge and the surfactant conferred a positive charge. However, in this case, the polymer did not confer a neutral charge. Next, we wanted to see if the DLS pattern changed over time. For this study, we analyzed and reanalyzed a series of silver nanoparticles. Particles were synthesized in November of 2020, and photographs of each sample are shown to the left. It's important to say that even as of today in late March, the colors of these samples still hasn't changed. Spectroscopic features are shown to the right. The dispersity, diameter, and surface charge values were similar in January as they were originally measured in November. For surface charge, the acid conferred a negative charge, the surfactant conferred a positive charge, and the polymer conferred a neutral charge.
So we have some immediate next steps that we are taking. So the synthesis mixture contains not only the target nanoparticles, but also unreacted reducing agent, stabilizing agent, and metal salt. So there's a variety of methods that we can use to separate the nanoparticles from these impurities, such as ultracentrifugation, nanofiltration, microfiltration, magnetic separation, although this only works for magnetic nanoparticles. And we can also use dialysis, which we have selected in this case.
Silver, copper, iron, and zinc nanoparticles were synthesized and coated with stabilizing agents to retain their shape and size, as well as to impart distinct surface properties. The properties of these nanoparticles could be exploited by the pharmaceutical industry.
Also, this has a large impact. Our future steps have a large impact. So this will allow us to determine the importance of isolating the nanoparticles and suspension from other impurities produced during the synthesis process. So we need to know whether or not it is our nanoparticles or the other things in the solution, in the synthesis solution, that is causing the change. So we will carry out the process of removing the impurities and then comparing each to see whether or not what's being put out would be put out into the environment or stuff like that, or any other place, is truly what we want to be put out there. And so this will be done by verifying the effects of the nanoparticles with and without impurities. Lastly, we would like to acknowledge first the environmental department at Baylor, also the U.S. Department of Agriculture, and the Gus Glasscock Jr. Endowed Fund of Excellence. We'd also like to thank members of the SAYS group, Dr. Bernard Zeckman, Dr. Michelle Nemec, and Dr. Edward Braswell. Thank you.
Awesome. Thank you so much, Jake and Denny. I appreciate your presentation. If anyone has any questions, please either ask them or put it in the chat. We'll give about 3 to 5 minutes as we did for the other one. Thank you, guys.
Thank you very much for your talk. Again, I thought it was very excellent. Could you go back to your data slides for me? The first set that you had?
This one or?
This one before that.
For this. This one?
No, you had some figures that, yeah. Did you have figures before, like prior to this one? No, okay. Maybe it was the first one that you said. You can go back to that one. I apologize. I think it was the one after the table.
This one?
Yeah, okay, there we go. That's what I was trying to look at. So those are just Okay. You were just looking at the molecular structure and like all the characteristics then there?
Yes.
Okay, I was just like confused because it's just kind of like went by quickly. I was like, wait, what's going on here? Okay. And can you go to the next slide for me? Yeah, one more.
One
Sorry.
more.
Yeah. Okay. All right. I just wanted to look at some of those again. Alrighty. Thank you.
Sorry about it going quickly. We had to move
No,
through
it's
quite a
okay.
bit.
You had a lot of information to present. It's hard not to just like fly through it all. So you're very limited on time. I understand.
Awesome. And then there's one question in the chat from Benjamin, Mr. Benjamin. It says, do you feel that the centrifugation step done to increase the concentration of the NPs adds any artifacts in as to how the NPs are behaving and aggregating? So if y'all would like to answer that.
If you're talking about this, or sorry, oops. Let me get to the slide you're talking about. Sometimes a centrifugation could add or remove an artifact. For this specific time, we did not use a centrifugation method. I believe we mentioned this. We boiled off the extra water, so that way we just got rid of the water and we did not add any and we did not precipitate out any artifacts. But as to see how it aggregates, that's why we took the DLS again, so we can see. So you can see the size of the nanoparticles can be slightly affected. This show right here in this middle graph, you can see the slight effect on. However, they're still, in the right range, so we're not too worried. And there's does that answer your question, Ben?
Mute. I'm sorry, I had to unmute. I thought that y'all were saying that to increase the concentration, you were doing some sort of centrifugation thing, but maybe I misunderstood. So
there's two methods you can use. You can use centrifugation, but we used the other method. We just mentioned both.
I guess I misheard then. Sorry.
We have another question. How do you expect these particles to be used by pharmaceutical industry in the future? So there's a couple ways. So one way that they can be used is they just have inherent antimicrobial, antiviral properties. So one way that they have been used already, I believe, and it was going through testing earlier last summer, was they were adding these nanoparticles onto like masks. So it's not really truly like the pharmaceutical company, but they were adding it onto masks so that way it would prevent the buildup of viruses and like prevent the growth of bacteria on the masks, which then had to be reused. Also, these nanoparticles can be used as drug delivery carriers to help make sure they get to the right spot in the body. Does that answer your question, Ashley?
Do we have time for another question?
We have
I saw it. Never mind. I saw your comment. You're good.
Yeah, sorry about that. If y'all need extra time for the judges just to continue to fill out the paperwork that you have, just tell me. But if you are all ready to go, then we will move on to the third presentation. So our third presentation is by Cassandra Gamez, and she is from UT Arlington. Cassandra, you can begin to share your screen whenever you are ready.
Okay, awesome. Does that show up for everybody?
Yes.
Alrighty. So my name is Cassandra Gomez. I am an undergraduate student doing my bachelor's degree in civil engineering at the University of Texas at Arlington. And today I'll be presenting the work I've been doing this semester over growing awareness of environmental quality in artisanal small-scale gold mining communities through youth environmental education. And this is a project that I'm doing under Dr. Smits and in partnership with the Responsible Mining Resilient Communities program that's funded by the National Science Foundation. So just a quick breakdown of the presentation. I'll go over some project background, my research question, the restrictions I identified, the methodology that I used to complete this work, my material development, overall outcomes, and then the time for acknowledgements and questions. So for some background, Colombia is an extremely biodiverse country, which heavily depends on paramos, which are Andean grasslands, as a water source. Unfortunately, factors like illegal mining, deforestation, and drought are negatively affecting these water sources and have even decreased amounts of usable water. In Colombia, mining activities are popular and very widespread. The sector contributes about 12 to 13% of the world's gold and directly employs millions of people. Now, this field wouldn't present as many issues if the practices were more regulated. It's estimated by a report on transnational organized crime that up to 80% of mining practices are done illegally, so they're not following the country regulations. And when gold mining activities take place illegally, pollutants like mercury and cyanide are used and often improperly handled, improperly disposed of. So they're able to enter the environment and contaminate the air, contaminate the water. Mercury tends to accumulate in fish, which then causes issues for people who eat the fish, neurologically, damage to organs. So it's an ongoing issue. And it is important to note that these practices have a large effect on local economies. So it's unlikely that they'll ever fully stop these practices, but adapting them so they're less impactful is important. And for this, community partnerships and education are crucial. One of the regional environmental authorities in Andes has identified difficulty implementing an environmentally conscious culture as one of the largest issues with continuing real lasting change in this area. So to address all of that mentioned, I worked on a youth environmental education program, and we chose the middle school range so that we could target school age students, and also because it's the average education level. So this material can be translated even outside of the classroom to members of the community. And then using all of this, my research question that I developed is what environmental education program could be added to existing school curriculum in Colombia in efforts to increase student awareness of environmental stewardship. So that will be the focus of my research going forward. Some of the restrictions I considered going forward with my research are, again, that mining is a multifaceted issue. And since it's so beneficial to a lot of communities, creating a dialogue that would paint it in a negative way would be detrimental to the relationship that we have with these communities. We work with Santa Rita and San Agustin right now. And then additionally, the activities that we wanted to include in these learning materials, we want to use materials that are not finite. So we want to be able to include things that we can reuse to decrease waste and increase longevity. We're ensuring not to overlap with existing mining education programs, such as the Mines Cares, which the Colorado School of Mines does. And topics covered will also be within the Colombian middle school curriculum. Additionally, some cultural and social barriers will be kept in mind. For example, the need to translate these materials to Spanish, which is something that I will be taking on because I'm bilingual. And I also made sure to avoid painting other very region-specific activities, such as coffee plantations, in a negative way, while still touching on the effects of fertilizer runoff on the environment. So a broad overview of how I went about this. I started with reviewing Colombian curriculums and lesson plans to really see what was being taught in schools, to get a feel for my scope and some of the topics that I wanted to focus on. And then I went in and I did more research on topics that I thought would be interesting to middle schoolers, and something that could be really interactive and something that would help build that connection to the environment. And then with all of that information, I built a lesson plan around those topics. I created some activities for the students. And these materials that I have prepared will be given to those communities in Colombia to be used by the school. The softwares I use are Global Mapper, SketchUp, Cura, and Google Earth. So after all of that, the broad topic that I wanted to focus on was the relationship between land use and water quality. It's something that's easily applicable to the region, and it can be a really interesting topic. Some secondary topics within that will be an understanding of what watersheds are, understanding what water quality is, and then the link of how land use fits into all of that. So how land use affects water quality and how land use is influenced by watersheds. And then additionally, we will be discussing point source and non-point source pollution within those materials. So the first activity that I prepared to tie into the watershed discussion is a demonstration of how watersheds work. And I wanted this to be something that was interactive and attention grabbing for the students. So I wanted to use a local example. And what I did is I went to the community of Santa Rita and I found a watershed that runs right through the middle of the community that contains the river that they use as their water source. And I modeled that. So the image on the right is where the community is located. The image on the left is the model that I created. Using Global Mapper, I got the elevation data for that, and then I put it into SketchUp and I enclosed it so I could create a 3D model. From there, I exported it to Cura, and I was able to print it. I actually have a small version of the one that I made here. It's kind of hard to see because it's white. But yeah, you can see the contours of that. And what I plan to do is expand on this and build a base and create a drainage system so that we can actually run water through it. We'll put food coloring on it to simulate different pollutants from different types of land uses and how those come together to affect the same water source. And then I'll be reprinting it at a larger scale as well for them to use. And then a second activity that ties into the water quality focus is one that I created that would allow us to avoid using disposable testing strips and to avoid having to buy expensive equipment. So I created a lesson where students could use observable parameters of water, such as the color, presence of vegetation, aquatic life, presence of macroinvertebrates. This color can tell a lot about the water. So the key on the left would be used during the activity to help identify what exactly is in the water. And then observations of varied and abundant vegetation and aquatic life would point to healthy water quality. And finally, certain macroinvertebrates can be identified to estimate levels of pollution in the water, because some are very tolerant of pollution, some are very intolerant of pollution. So that would tell you what the water's like if they're found in large numbers there. For example, if you found a lot of stonefly nymphs, you'd know that the water quality is likely good. And then a key to identify these microorganisms will also be provided to the students so that they'd be able to identify what exactly is there. And an example of how they would fill out this table is here. So if they observed that the water had a green tint to it, then they could assume that that meant phytoplankton and algae in the water. If they found a lot of right-handed snails, pollution levels are probably low because they're very sensitive to pollution. And then observations of varied and abundant life, vegetation, and aquatic life would point to water quality being healthy. So this activity has been developed to help them get an overall picture of what the water quality is without even having to test it, and just by using what they can see, and to learn to use those tools that they have. In addition to the activities, some discussion questions are provided throughout the lesson. I wanted to use these to keep the students engaged and to focus more on their interpretation of the topics and encourage them to think of examples in their own life. So these questions will help students to understand the material and ideally continue to think about it as they move forward with their education and in their lives. Although it's difficult to quantify the outcomes of this project with concrete data, Some of the outcomes that I'm aiming for with this project are that the student will thoroughly understand the material, meaning that they'll understand how pollutants move through the environment, how they impact the environment, and doing by continuing the conversation with other students, teachers, members of the community, their parents, etc. It also involves students being able to identify specific sources of pollution in the world and in their community, and learning to learn just by having the discussions in small groups during the lesson and having students actively participate in them. And that concludes my presentation. I'd like to quickly thank Danielle Cambarellis for providing me guidance through this process and for helping me with my research question, and my mentor, Dr. Smits, for providing me constructive feedback and always helping me to think of problems through different lenses and perspectives, as well as all of you for being here today. So with that, does anybody have questions?
Thank you, Cassandra. Yeah, if anyone has questions, feel free to either type it in the chat or ask in person.
Hi. Thank you very much for your talk. I thought it was really excellent. I love using the EPT taxa approach to helping students understand water quality. It's probably one of my favorite outreach things that I've done in the past. I really only have one question. It might be kind of silly, but your presentation title, I seem to have missed it. What is ASGM?
Oh, artisanal small-scale gold mining.
Okay. Yeah, I'm
I
sorry.
don't
I
even
was just
know
like,
if I said it in the beginning. I meant to. I might have missed that.
okay. All right. Awesome. I really like your ideas that you have here, and I think that it is very important, and it's something that I like your stuff. It's very good.
Absolutely.
And I appreciate the like the extemporaneous delivery too. It was very good.
Thank you.
So I have one quick question. So you printed a 3D model of the watershed.
I did.
Are you going to be printing, like, how large a scale are you talking about when you talk about reprinting that for them to see?
We don't want anything huge because we are going to have to take it over there. I'd probably say, oh gosh, maybe like 12 inches by 12 inches. Nothing tremendous. It has to be portable.
Okay. That was really my big question, because I know that I've dealt with 3D printed stuff before, so I know that you can reuse them and you can wash them and do like a whole bunch of stuff with them. So I really like the sustainable aspect of your lesson as well, because I think that's a nice touch that kind of makes, hopefully will make them consider their own impacts as well.
Yeah, absolutely. Thank you.
Hi, thanks for that presentation, and that was interesting to learn more about Colombia, especially. I didn't know a lot about that with the mining that they do there. My question was kind of like on the context of like, where do you think your work fits in? Like, is this one of the first initiatives to really do youth outreach and bringing awareness to them, or has stuff like this been done in the past? And how does your effort differentiate, or what kind of outcomes do you expect would be different? Because I'm sure mining's not new and we've known for a long time that it's a problem. So I was just curious what you thought of that.
Right. So there are some broad initiatives, but we only work with 2 smaller communities that are pretty rural. And then the Mines Cares program that I mentioned, they focus specifically on the issue of mining. But we wanted to do something that wouldn't directly say, like mining is an issue. We wanted it to be, because we want a lot of community engagement, because like fixing these issues really has to come from within. It can't be us coming in and saying, mining is bad, you need to fix this. It has to be like creating an awareness and then wanting to find solutions on their own. So I think that's what differentiates this program.
Okay.
Awesome.
Do we have time for one more, or are we?
Yeah, we have about a minute left. Yeah.
Okay. Is there? I've been reading about Bitcoin mining. This is the word mining here is just a copy word. It has nothing to do with anything in the ground or the environment, but it's very like energy intensive. And I think in the next few years, people are going to become more aware that like it's like extremely, extremely draining in terms of like, I think there's like more like Bitcoin uses more energy than like Finland and stuff. And I was wondering if like any of the paradigms you have could be translating to other future emerging environmental problems, especially when it relates to like the economics like gold and then, you know, Bitcoin and things like in the future. And kids like these kinds of new avenues, but might not be aware of the environmental issues.
Right. I think that's definitely true. I think we have to be conscious when things like this emerge to discuss all the sides of it, because there might be benefits, right? But you have to ensure to tell people that there can be a bad side, and we have to be conscious of that. So something like this, where you're teaching it very broadly and talking about a lot of different specific topics, I think can be applied.
Thanks.
Awesome.
Thank
Thank you so much, Cassandra. Thank you for your presentation. Up next, this is our last presentation of the day. We have Harrison Coker from A&M. Harrison, you can begin to share your screen whenever you are ready.
Thank you. Oops. All right, can we see that okay?
Yes.
All righty, sounds good. Hello, everyone. My name is Harrison Coker, and I am a research assistant in Dr. Bradford Wilcox's ecohydrology lab at Texas A&M University. I am graduating this semester with dual degrees in renewable natural resources and forestry, and today I'll be presenting to you about the ecohydrology of the post-oak savanna. So the terrestrial water cycle is essentially driven by precipitation, and precipitation is what's going to provide biologically available water to the soil profile for uptake by microbes and by plants and other organisms that are living within the rhizosphere. However, a significant and often overlooked portion of rainfall is often lost in what is called interception by the plant canopy. And there are many factors that can influence canopy interception losses, including canopy storage capacity, leaf area index, water repellency, and projecting tree crowns. But this is interesting because oftentimes when we're trying to measure the total evapotranspiration of an ecosystem, we are not directly concerned with the evaporative losses from the plant canopy and the reductions in throughfall to the soil surface. And this is significant because in rangelands that are becoming encroached with woody plants, there might be significant changes in the amount of water that's actually reaching the soil surface. So in the post-oak savanna of Texas, we have a biological community that is considered to be structurally unstable. And what that means is that we have woody plant encroachment in the form of evergreen shrubs moving along fence lines and in the subcanopy regions of climax trees. And we can see that these shrubs often dominate within 5 to 10 feet of the forest floor. And so as we are seeing a landscape scale transition as a result of overgrazing and lack of fire regeneration, and as essentially a negative consequence of human interaction and overexploitation of the environment, we're really not sure how this is affecting the local water budget and if this introduction of woody vegetation can significantly reduce soil moisture conditions that may ultimately lead to deep groundwater recharge and local stream and aquifer recharge. And the post-oak savanna is an interesting place to study this because of the underlying geology and the sandy clay soils. So just as to include a picture of Yaupon holly here, which is the primary vegetation investigated in this study, you can see that the region of forested sites and what would otherwise be a native grasses and large oak trees is really becoming dominated by these monospecies clusters of Yaupon. And this is often evaded most ecological assessments and is something that's primarily occurred over the last 100 years. And so our central question is essentially, does Yopon invasion significantly alter the hydrologic conditions of the post-oak savanna? And so to do that, I have, for my undergraduate research, trying to determine interception losses of common post-oak savanna shrubs and determining near surface soil moisture dynamics under shrub clusters and open savanna, and comparing these two to see if there are the changes noticeable or significant. And so moving on to my interception method, I created around 50 or 60 of these, what I'm calling through-fall devices, and placed them under the subcanopy region of our shrubs. And after every rain event, I got to crawl around on the floor, and I measured with a graduated cylinder the amount of volume that was reaching the soil surface. And that was compared to an unobstructed rain gauge that was located away from any vegetation. And then these were compared, as I'll show in just a minute. And then to understand soil moisture dynamics, we had two different methods. Continuous soil moisture sensors were installed at 3 locations, 2 under shrub canopies and 1 in a grass site. And so we dig an excavation pit as seen, and we install time domain reflectometry sensors that take a measurement of soil volumetric water content every 10 minutes. And for discrete soil moisture, we are using what is called a neutron probe, as seen here. And that is going to provide the gold standard for soil water conditions at varying depths within the soil surface. The neutron probe is essentially placed on top of an access tube, and the soils are characterized on the way out so that we can calibrate and have an accurate estimate of soil water conditions. So here's my interception loss survey. I am right in the middle of getting all of my data together for my manuscript. So this is in the works, but essentially here are the different monitored canopies. And here's just a kind of picture of Eastern red cedar and Yopon so that you can get a glimpse, but these are not the canopies that I monitored. But essentially we're seeing interception losses to the subcanopy floor extremely high under juniper or Eastern red cedar, excuse me, and Yopon holly, which is similar to other studies that have been performed across the southern Great Plains and in other semi-arid climates. And Yopon holly is ranging from about 21 to 36% interception loss. That is very significant. It's more significant than semi-arid shrubs. And we're seeing these essentially shrub islands or mixed species assemblages coming in somewhere just under and below that amount. But these are all very high interception losses. We are essentially seeing a very significant portion of precipitation not reaching the soil surface because of these woody shrubs. And so here are all of those groups again. Each of my data points here is the average of the through-fall collectors per precipitation event. And so we can see that typically for each of our rain intensities, we have somewhat of a logarithmic trend line. These were just included for visualization purposes. They're not significant or anything, but the data essentially presumes that at lower precipitation events, as we see on the x-axis, the interception loss as a percentage of total precipitation decreases with increasing precipitation events. And so the canopy storage capacity, or the amount of water that can actually be held in the plant canopy, is more efficient at lower precipitation events where there's less stress to the plant canopy. And so for our soil moisture conditions, I unfortunately do not have my 40 centimeter sensor working under my second mixed shrub canopy. But essentially we're seeing rather similar volumetric water contents under both shrub canopies and in grassy savanna. And at the location that I am measuring, we have an assemblage of native grasses, big blue stem, and some others. And it's rather interesting. At the 40 centimeter profile, we are seeing that the shrub cluster has reduced volumetric water content. But eventually this data will be put together and we will be able to understand the amount of water held in a column of soil and compare that across sites. And unfortunately, the neutron probe data is not processed yet, but that is going to be extremely clear in what is occurring across these sites as we can compare more woody to grassy covers. And so all of these data are being monitored through April. And we are essentially seeing for the first time published results of Yopon, intercepting between 21 and 36% of precipitation. Interception losses for eastern red cedar are similar to those of other studies. And we are seeing that around the 40 centimeter region, we are having reduced soil moisture conditions, which could essentially offer a competitive interaction hypothesis for why woody plant encroachment is so successful and is proliferated so with such a high frequency across the oak savanna. And so with this understanding, especially with interception losses, we can infer that there is a community level loss of interception or precipitation that is occurring. And this most likely does have very substantial implications for how much water is being recharged to the Carrizo-Wilcox Aquifer, and for future studies, we'll be able to essentially use this data for further modeling and to better understand some of the dynamics that are occurring. And here are some references that I have. And here's just a picture of Post Oak Savanna taken at Independence. And thank you all for listening.
Awesome. Thank you, Harrison. If anyone would like to ask any questions, please feel free to ask him out loud or put them in the chat.
So well done.
Thank you.
I guess, so for Yopon holly, if we know that there is an interception loss now, is there any known way to control the holly? I know, like, when I went to Clemson, they did like controlled burns for invasive species, and we do that here at the wetlands in Waco. Is that something that is effective for Yopon holly? Is it an option for the area that you're seeing these monocultures in?
So fire is unfortunately socially unacceptable across most of Texas savannas. We don't see fire successfully occurring like we do in the greater prairies of North America. But Yopon holly in particular actually is very good at reducing fire, and prescribed fires that occur at the Ecology and Natural Resources teaching area here at TAMU show fires that will sweep through grassy regions with destroying the fuel load, and then they will get to a shrub canopy and they will stop. And the other problem is that when we mechanically remove these, they have extremely high resprouting capacity. And so removal of this is typically done with chemical treatment, and that is, I think, for landowners, really the only successful way of going about it.
Thank you.
You're welcome.
I echo that this was a great presentation and really clear. So good job on that. My question is, like, is there some kind of carrying capacity that this gets to with like Yopon invasions or anything eventually? Because surely their root structure doesn't go as deep as the other, big trees, and won't they eventually deprive themselves, or, some sort of negative feedback there? You kind of hinted in your conclusions, but I was just wondering what if this just keeps going unabated? What happens eventually?
Thank you. Great question. So the arguments of woody plant encroachment are actually the focus of the lab that I work in, and that's been an ongoing debate for a number of years. But what the thought is that a savanna in transition or undergoing continued transformation could potentially really limit groundwater recharge. And also, we are starting to believe that there are more, or I'm starting to believe just from my time, the years that I've spent observing this, uh, believing that the actual ecology and biochemical cycling of these regions is going to be more significantly altered. It's actually appearing that despite Yopon intercepting such a high amount of precipitation, there doesn't seem to be a depreciated soil moisture conditions. And I think a big part of that is because water is being held in the litter canopy. And so I don't think that there's necessarily any thresholds for this kind of encroachment or anything that we should be looking for that would be detrimental to us as a society, but rather understanding more complex processes like carbon loss from the foliage that's stored under Yopon canopies and held in the litter, and how much of this water is being transpired, being sent back up to the atmosphere, I think would be very important for like local humidity conditions. But other than that, the arguments are pretty unclear.
Okay, thank you.
You're welcome.
Awesome. We have about a minute left if anyone else wants to ask a question or has a comment.
I don't have a question, but I just wanted to say that was an excellent presentation. Like, very good. And I could tell that you're very knowledgeable and passionate. And I think that that's really nice to see, especially in undergraduates, that they are, have put a lot of time into really understanding their research. And, the way that you answered both Ben and Ashley's questions just definitely showed that you really care about your research. So just want to say good for you.
Thank you so much. I do want to point out this is a sweet gum, liquid amber styractiflua. It's a local savanna species, but you can tell that the interaction of rainfall from the plant canopy is really a significant driving force that is often not understood or really very well researched, despite actually dominating where we are attributing most of our water. And so as this modifies the chemistry and the actual quantity of through fall reaching the subcanopy zone, this is, I think, an important study that can really be expanded very successfully, and further information can be built upon.
Awesome. Thank you so much, Harrison. I appreciate the presentation. Thank you guys for coming. You all did a wonderful job. This now concludes the Environmental Science Division. However, we're going to take a picture of everyone if I can figure out how to, just for social media purposes. So please turn on your camera if you would like to be in it. Let's see. Okay, I'll take a screenshot with this. Okay, Control Shift. Okay, and then let me take one picture on my phone just in case. All right, so sounds good. Y'all are free to go now. Judges, if you would like to stay and deliberate, go through all of your notes with one another for a little bit. Whenever you are done, we have more events for the rest of the day. Those events begin actually at 11:15, but it's okay if you're late to that. And yeah, I appreciate y'all coming out. Again, this is my first time as a moderator, so thanks for being patient with me, and y'all have a great rest of your day.